class, you will have a significant results. Mas measurable siya. Kasi hindi naman natin ma-measure yung number of microorganism present dyan in terms of their different concentration. Kung tanaw lang na to siya, di ba? So, para maging reliable, significant, measurable ang ating data, we're going to plate that. So, ato siyang i-pour out sa plate. So, ayan, nakikita ninyo na, i-pour out na siya. So, after that, we will going to incubate that. So, after natin ma-incubate, no, there will be microbial growth. So, ang gagawin dyan is, hindi mo na siyang i-count, no? Hindi mo siyang i-count kung how many colony formation or microbial growth na nag-occur dyan. Pwede siyang i-count manually. Pwede naman din with the help of the colony counter, no? So, pero, meron din ibang studies class na hindi or experiment na yan. Dili na siya colony counting, no? Meron pang iba. So, example lang yun. You're going to determine as to the number of colonies formed after na incubate na with the method using pour plate and serial dilution. Ayan. So, sa pour plate, nakita natin dito sa baba, it involve mixing. So, dito class, hindi ka maggamit ng hockey stick. Ang gagawin mo is, after mo siya na-pour out yung sample natin dyan sa petri dish mo, takipan mo na siya, and then what you're going to do is to use your point finger, place it on the center of the lid of the petri dish, tapos imo siya i-move circularly. Gently lang kayo. If ever na plastic ang gamito na petri dish, so pwede siyang mabuak. So gently lang. Until such time na ma-occupy niya talaga ang buong petri dish. Ayan. So, ito na yung serial dilution natin. So, it's a series of sequential dilutions used to reduce a dense culture of cells to a more usable concentration. Each dilution will reduce the concentration of bacteria by a specific amount. So, try to notice the arrow class. Ang first test tube natin, it is considered to be concentrated. And the original inoculum is nandyan nakalagay. So, SOP yan siya class sa procedure sa serial dilution na we will have 9 ml of broth or pwede naman din distilled water. Ayan. Then, we will have 1 ml of original inoculum for our first test tube. So, dapat mix-mix na siya. Dapat meron ka talagang specific stirring rod dyan. Hindi pwede na isang stirring rod tapos gagamitin mo sa the rest of the test tube. Hindi yan siya pwede kasi it will make your data inaccurate. So, dapat merong specific stirring rod. So, mix-mix dyan. And then, after that is, ang 1 ml sa first test tube is we're going to put that sa second test tube and so on and so forth. So, in order for us to appreciate yung number of microorganisms present doon sa ating test tube with the different concentrations of inoculum, is we're going to have plating, So, ato na siyang i-pour out dira sa ato ang dish. Tapos incubate, after incubate, that's the time that we will identify na dili good sila pareha o growth. So, sa first test tube nato, since na-adra atong original inoculum, so therefore we can say that the microbial growth is pretty different compared to the rest of the microbial growth, Kasi siya man ang original, so siya gid ang pinakadaghan na colony formation or microbial growth, Mas taas, mas massive ang iyang microbial growth. Ayan. We can also use serial dilution class as spread plate technique. So same pa rin, in a specific test tube class, meron din siyang specific na petri dish, Ayan, na merong nutrient media. Tapos gagamitin mo yung hockey stick para spread out yung sample evenly, then incubate, then observe. Ayan, tapos na tayo kay inoculation, incubation, then isolation, no? Ang taas kay isolation, no? So next is inspection. So naka-isolate na ta. So ang gagawin natin is i-inspect mo na siya. So ang gagawin dito sa inspection is you really need to examine the growth, macroscopic and microscopic, no? Hindi lang po na simple kitanaw lang nimo siya kung sa imo makita si muhang mata, but you need also to examine it by use of microscope, no? So you need to describe, to identify, no, appearance ng colony, size, shape, texture, color, lahat. Pwede din class na you will also subject your sample class into staining para mas ma-appreciate mo pa talaga yung identification, I mean yung examination mo sa sample mo, no? So ang last I is ang identification. So after mo na-examine siya with all those different things that you need to examine sa specimen mo, is you're going to identify na unsa ba gyud niya ako kung nakitaan si Bacillus anthracis ba ni or unsa ba ni si Staphylococcus aureus ba ni, ana. So before you will conduct your experiment class, it is good that you will also need to research ano yung information na meron or ano yung required information that you need to know before conducting such experiment. So dapat alam mo kung ano yung characteristics ng microorganism that you need to study. Alam mo kung ano yung hitsura niya para pag identification mo is alam mo na kung okay, mao din yung hitsura, mao na ni siya, good. So but aside from that class is you need to know the characteristic, the morphology, which refers to the structure, no? Is aside from that class, there are also biochemical tests na which helps you to identify such microorganism, no? So meron tayong mga different biochemical tests na that would help you identify na mauna ni siya na microorganism yung ginapangita. So therefore, kung ikaw nagkanda ka ng experiment class, dapat po talaga ready tayo before conducting the experiment. Dapat naka-research na tayo para alam ba natin kung ano at ano ang meron doon sa ating hinahanap. Baka naghahanap tayo pero hindi natin alam kung ano yung hinahanap natin, no? Ayan. Okay. So next is media. So, alam na natin no, na si media, yun yung medium natin that will provide nutrients to our microorganism which we isolated sa laboratory. So, ang media natin is considered to be extremely varied in nutrient content and consistency because ang media natin is iba-iba. Each media has its own ingredients, no? Iba-iba yung ingredients nila, iba-iba yung purpose nila. So that is why na they are extremely varied in content at saka consistency. Tapos ang media class mostly it is designed for bacteria and fungi, no? Na pag-isolate ng mga microorganisms. Aside from that is yung culture medium natin class is considered to be solid, liquid, no? Or liquid preparation used to grow, transport, and store microorganism. So later we will identify this different medias class na ano yung mga different media class in terms of we're going to grow this type of microorganism, ano yung media na gagamitin natin if we're going to transport a microorganism coming from remote area or etc. Aside from that is much of microbiology depends on the ability to grow and maintain microorganism in the laboratory. So mamaya din makikita natin yung different medias natin na if ito yung gagamitin mo na media, ito yung effect sa isolated microorganism na na-isolate natin, no? So it comes also with the objective of our examiner. Then, microorganism's normal habitat reflects its nutritional requirements. Of course, kasi yung nutrient media natin, that will be the source of nutrients na ating microorganism. So, magiging habitat niya talaga yun. So, dapat nandoon yung necessary nutritional requirements ng ating microorganism. The next is ang uses. So, first is to identify the cause of infection from the clinical sample so that proper treatment can be given. So, if ang doctor or ang pathologist want to study this particular microorganism, it helps them also to study more, no, if mag-culture sila nito na microorganism. So with that, they can administer proper medication, diagnosis, etc., etc. Next is to study the characteristics or properties of microorganism. Yeah, same goes with number 1, that enables them to identify, ang microorganism, more information about the microorganism. Then next is to prepare biological products like vaccine, toxoids, antigen, etc. So, just like sa nangyari sa COVID-19 class, di ba pag the time na we are already on pandemic, yung early time pa talaga ng pandemic, wala pa talaga, di ba, tayo ng vaccine. But with the help of science and with the help of many researchers class, they were able to come up with vaccines, no? So just like yung sinabi ko before na, just before pandemic hit is meron na talagang other studies na na-conducting scientists. Nag-progress lang siya even during nag-hit yung pandemic, no. Mas nag-advance yung studies nila, yung knowledge nila in terms of what is coronavirus, considering also the different variants. And since ang daming variants natin, it helps them also to understand na yung ang characteristic ni coronavirus, na sila similarities, tapos sila differences, no? So sa nutrients naman ng media natin, basically they have the water. This is very important for our microorganism in order for them to survive. Kaya kung wala water, mamatay na sila, no? Especially doon, di ba, sa cell nila, sa cytoplasm nila, it is considered to be a water-based environment. So dapat, hindi siya dissipated ang microorganism. Then we also have carbon, nitrogen, buffer system, and other minerals necessary for their growth. So ito na yung part natin sa media class that we're going to classify them in terms of their physical states, chemical composition, and functional type, or in terms of their purpose, no? So for physical state class, like for example, the liquid media. So itong liquid media class is considered to be water-based solution that do not solidify at temperature above freezing that tend to flow freely, of course, from the name itself, no, liquid. So this liquid media is also termed as broths, milks, or infusion. So ang manifestation ng appearance nila is throughout the container, dispersed, cloudy, or flaky. Then sa examples natin, class, every type of media class, I will give examples, but don't worry, I will not dig more on the information of each example sa media natin. I just presented some of the examples here. So examples natin sa liquid media, we have the nutrient broth, methyl blue milk, and litmus milk, and fluid thioglycolate. Sa semi-solid naman class, if before, palagi kong ginamention si solid media, aside sa liquid media na nasabi ko kanina, is we also have semi-solid. So, hindi talaga siya katulad ni solid media na solid. Pero ganito yan siya class. So, solid media class, kung tanaw na mo siya sa Petri dish class, dili po ka makainom na solid good as in literally solid, no? Kasi para din siyang gel-like class, pero mukuan siya, mura siya, jellyase, yan, ganon. But considered as solid, no? Muna ilahang definition sa solid media. Sa semi-solid naman, it exhibits a clot-like consistency under ordinary room temperature. So meron pa rin siyang presence of solidifying agent, no? So yung solidifying agent natin na meron sa solid media and semi-solid media class, yung component na meron class kung bakit siya nag-solidify is ang iyahang agar class, atong pinaka first step with class nga mag-timpla ka class para to come up a media. It is what we call as agar. It is a powder-like which is you're going to prepare. Kumbaga, mura kang nagtimpla o juice class. So with that agar class is ang main component kung bakit mag-solidify ang media is isang active ingredient niyan is ang red marine alga. So that is the main ingredient of our agar kung bakit siya mag-solidify. Ayan. So aside from that is ang semi-solid class is used to determine the motility of bacteria and to localize a reaction at a specific site. So if you are studying more about the motility of the bacteria, for example, di ba na-mention natin before yung flagellar staining technique, no? So before you will come up with flagellar staining technique is what if nag-isolate ka of bacteria? And then, for example, mixed culture ang bacteria mo and you're using a semi-solid media. So what you're going to do is, sa semi-solid media, since hindi naman talaga siya totally solid siya class, it helps our bacteria to be motile. No? So kung magiging motile siya, mas easier sa iyo na makakuha ka ng, maka-isolate ka ng bacteria that could be subjected also to flagellar staining technique. No? Maybe because you want to determine the number, the presence, and the arrangement of the flagellum. Ayan. Then examples niya, motility test medium and sulfur indole motility medium. Then sa solid media naman class, this is good for isolating and culturing bacteria and fungi. That is why this is considered to be common, no? And then we have two forms, the liquefiable and then non-liquefiable. Sa liquefiable class, very unique ito siya kasi this is considered to be reversible. So pwede siya na solid to liquid and the liquid to solid. Then we also have the non-liquefiable. So hindi ito pareha ni liquefiable class ni reversible. So si non-liquefiable do not melt. At the same time, ang non-liquefiable class can be coming from different sources class. It could be from rice grains, cooked meat media, egg, or serum media. Meron kasi tayong media class na hindi siya synthetic. So mamaya may encounter natin si synthetic. Synthetic means na chemically produced siya ba, no? Or naka-powder na siya class, no? So, mas easier si synthetic kasi naka-powder na siya. All you have to do is to prepare, no? Pero meron din type of media class na para talaga siyang pinakbet class na ikaw talaga mag-prepare to come up a media. So, mas kapoy. Ayan. So, in terms of chemical content of media, meron tayong synthetic and non-synthetic. So, ito yung sabi ko kanina. So, si synthetic chemically defined composition, then it contains pure chemical nutrients and molecular content. Si non-synthetic naman yung sabi ko kanina na exactly opposite of synthetic that could be coming from different sources, no? Yan. Meron tayong other different sources, blood, serum, meat extract, infusion milk, etc. Then examples natin sa non-synthetic class, na pwede siya coming from the different sources class in order to come up with a type of media is nutrient broth, blood agar, McConkey agar. So ang last part is the functional type. So we will we will enumerate different medias class in terms of what is your purpose nga nung kana man imo kang gigamit na media, no? So kani siya itong functional type class, dapat din ito din yung kailangan i-identify ng examiner, yung scientist, yung microbiologist natin, na depende din kung ano yung scope ng kanyang study, no? So sa media class, meron tayong media class na specific talaga according to its purpose. Yun yung nasabi natin kanina, di ba? Yung intro sa media. Pero whether bacteria or fungi, no? Pero sa media class, meron din tayong a type of media class na pwede siyang general use. And that is what we call as general purpose media. So merong bacteria class na pwede siyang gamitan na general purpose media, hindi talaga siya specific. Like for example, yung nakanta ko na experiment before is a very specific media ang kailangan gamitin sa katong fungi. So, yung specific media class is hindi siya available dito sa Philippines, no? So, na-shock ko, pwede siya Philippines. So, expect na lang na mahal. So, I found out na it is available sa Singapore. So, doon ko siya na-purchase and then I need to also to get help from our laboratory stockroom. Kaya na sila regulation. So it needs to be, kailangan siya i-pass by pa sa custom, sa lab, mag-inform ka sa lab, kaya medyo yun siya, very sensitive na chemical. So, ayun po. So, we have a type of media na for general purpose. Ayan. So, it is designed to grow a broad spectrum of microbes that do have special growth requirements. So, aside from that, example natin na considered to be general purpose media, si nutrient agar, and broth, brain heart inclusion, and TSA. We also have enriched medium or media, no? So, aside from it comes from blood, serum, hemoglobin, si enriched media is also have special what we call as growth factors. Itong growth factors na meron si enriched media is very important in order for the species to grow. So therefore, itong growth factors class, meron siyang organic compounds that the microbes cannot synthesize themselves. That is why they need to have growth factors sa enriched medium para mag-grow si microorganisms. Aside from that, si growth factors is considered to be fastidious, no? So, kumbaga, mag-boost siya microbial growth sa ating microorganism. Example ng ating enriched medium is si blood agar coming from the sterile animal blood ng sheep. Then, next is si selective and differential. Overview information of selective and differential media. This is very, this is considered to be cleverest and most inventive media because of its purpose, no? Designed to have for special microbial groups. So, yung sabi ko kanina na cleverest and most inventive media siya because of its purpose class, kasi mamaya we are going to know na bakit ganon. Okay? Sige, unya na. So, si selective o si differential class, this is considered to be good application class if you're going to isolate and identify microorganisms. Aside from that, it also permits preliminary identification of a genus or even species because of the ability of this kind of media sa pag-isolate and sa pag-identify. Bakit? Si selective media, ang gagawin ni selective media is i-inhibit niya ang other unwanted microorganisms to grow. So kung si Bacillus anthracis lang yun ang imong ipag-grow, si Bacillus anthracis lang yun ang ipag-grow. Wala na ilain, no? So depende pa rin yan kung ano yung scope ng study mo, no? So ang selective media, it hastens isolation by suppressing the unwanted background organism and allowing growth of the desired ones. Examples, yung mannitol salt agar, mag-country agar, and hectone enteric agar. So differential naman is kung i-inhibit niya ang uban to have a single species lang talaga na mag-grow, dito kay differential is pwedeng ang dami. For example, ang daming nag-grow na bacillus pero different species. So ang edge lang aning differential media is you want to bring out the visible differences among those microorganisms. So moto siya ang iyang special features sa differential media. So for example, si Bacillus anthracis kay it's a kind of bacteria, di ba? For example, ang imuhang gusto na i-bring out na visible differences in terms of their flagellum ba, or in terms of their capsule, maybe, no? So, pwede mo yan siya na yun ang sabihan mo. It brings out the visible differences. So, paano nangyayari yung visible differences nila? Because of the type of chemicals, meron si differential media that reacts, that enables the microorganism to react, no? Kaya dagan na sila, di ba, dito? So, yun ang magiging effect niya. But aside from that, if you want also to have meaningful result, you can also practice staining technique, no? Kaya since i-bring out man niya ang visible differences, maybe you can also use staining technique para mas makita mo talaga, no? Ayan. Next is specimen transport media. Ito yung type of media na sabi ko kanina na if ever you will get microorganism or specimen coming from remote area, no? So ito yung gagamitin mo, so especially para ma-preserve yung ating specimen or yung microorganism natin. We also have the assay media. So this is very useful and common for our pharmacy students, no, or mga pharmacists. So especially if they want to determine the efficacy or if naabay antimicrobial, antifungal property ang isa ka plant, no? Ayan. So same goes with disinfectants, antiseptics, no? For example, we have different brands of antiseptics in disinfectant class. So you want to determine if sino yung pinaka-effective sa kanila. So therefore, you will be needing different microorganism in order to assess, to determine if anong effective na disinfectant and antiseptic sa kanila, no? So ang gagawin mo niyan, class, is sa mag-prepare ka muna ng assay media, and then ang gagawin mo dyan is mag-isolate ka ng specific na bacteria, tapos i-test mo na lang ang efficacy sa disinfectant or antiseptic ba, ayan. So, pwedeng ganun. So, sa lab, anak class, ang activity, anak class, if you're going to test the efficacy, no, the effectiveness of disinfectant, okay, magkuha ka different types of disinfectants, tapos imuhang i-assess ang microorganism kung asa siya most susceptible na disinfectant. So, lingaw po. Next is enumeration media. So ito siya, this is very effective for industries, environmental microbiologists na that they need to know, determine the count of numbers of organisms present sa milk, water, food, soil, and other products or samples. Okay. So medyo marami class, no? Hindi ba tayo na information loaded dyan? So is every questions class? Meron ba tayong questions class or clarifications about our topic? Okay. So far, medyo okay pa. Pero medyo nag-over information loaded lang tayo siguro, no? Tama ba ko? Nag-information loaded tayo. Kindly react naman dyan, class. Kasi hinihingal ako nag-lecture.